morning one and all. Now I would like to say about Unit 2 coral reefs and which we go see about phylum Porifera and phylum Coelenterata. Now let us see about a coral reefs. And a coral reef is an underwater ecosystem characterized by reef building corals. Reefs are formed of colonies of coral polyps held together by calcium. Coral reefs are made up of colonies of hundreds to thousands of tiny individual corals called polyps. These marine invertebrates animals have hard skeletons made of calcium carbonate, which is CaCO3, and are sessile. And these are the spicules in sponges. When we observe the sponges, we have some tiny spine-like structures are present all over the body of the sponges that we are called as spicules. And these spicules are of different types like monoxon, triaxon, tetraxon, polyoxon, which is depends upon their shapes and corners of their spines. It is called as spicules in sponges of different types: monoxon, triaxon, tetraxon and polyoxon. Next, most sponges produce skeletons formed by spicules, structural elements that develop in a wide variety of sizes and tri-dimensional shapes. The morphologies of spicules are often unique to clade or even species level taxa, which makes them particularly useful in taxonomic assignments. Besides supporting sponge cells, spicules can help larvae stay beyond while in the plankton or reach the bottom at settlement, enhance reproduction success or catch prey. Let us know about the types of spicules. There are three types: calcareous spicules, siliceous spicules, megasclids. And the calcareous spicules means the organic material in this type of spicules is calcium carbonate or calcite. And next, siliceous spicules, the organic material in this type of spicules is colloidal silica or silicon. Megasclids, these are large spicules constituting main skeleton of sponge body. Next, let us know about the important question: various canal system in sponges. When we observe sponges, their canal system depends upon different types. So like that, let us see about various canal system in sponges. This is Ascon type of canal system. This is Sycon type of canal system. In this Ascon type of canal system, actually observe these present at the top of the region is called as osculum, and the entrant canals, these holes are called as dermal ostia. Dermal means which is present outside the body of a sponge is called as dermal ostia. The holes present out the layer of the external layer of the sponges is called as dermal ostia. And these are the chenocytes. These yellow body structures are called as chenocytes, which are flagellated cells. In this we can observe flagella, which helps to the movement of water, which helps in the water currents. And the middle part of the area in the structure is called as spongocoel. The first layer is called pinacoderm, middle layer mesenchyme, and third layer chenoderm. In this dermal ostia, water ingresses into the water through dermal ostia, ingressing water through dermal ostia. Water enters into sponges through dermal ostia through this way and comes out through the osculum canal. This type we can observe in leucosolenia. In leucosolenia sponges we can observe this type of Ascon type of canal system. Next, Sycon type, similarly, but in this type in this type of cyclone, we can observe chambers. These specialized structures called chambers. In these chambers, chenocytes are present, which are flagellated cells help in water current. Same process like water current comes through the dermal ostia, enters into the chenocyted chambers, and through apopyle, the area present between the incurrent canal and excurrent canal is called as apopyle. Water enters through apopyle into the spongocyl, and outgoing water through osculum. Exits through the osculum. And this example, siphon. Body walls of cyclonoid sponges include two types of canals, the radial and the incurrent canals, paralleling and alternating with each other. Both these canals blindly end in the body wall but are interconnected by minute pores. The canal system helps the sponges in nutrition, respiration, excretion and reproduction. The current of water which flows through the canal system brings the food and oxygen and takes away the carbon dioxide, nitrogenous wastes and feces. It carries the sperms from one sponge to another for fertilization of the ova. Next, let us discuss about polymorphism. Some few coelentrates such as hydra and sea anemone are monomorphic, where only polyp phase is found, but other coelentrates display polymorphism. Polymorphism is the existence of multiple types of structurally and functionally diverse individuals within a population. The phylum Coelentrates class Hydrozoa contains a huge number of colonial species with several forms of individuals known as zooids. Polyp and medusa are the two primary zooids found in coelentrates. Let us know about polyp. Polyps in Hydrozoa has a tubular body with a mouth at one end encircled by tentacles. The other end is normally blind and linked to the substratum through the pedal disc. The majority of polyps are sessile. They undergo asexual reproduction. Next, let us know about medusa. Medusa has a bowl or umbrella shaped body with peripheral tentacles. And a mouth centrally positioned on the lowest concave surfaces projection called manubrium. Medusas move about often. They undergo sexual reproduction. Next, importance of polymorphism. It is necessary for the division of labor among the zooids. Distinct forms are assigned different functions. Polyps, for example, are focused on feeding, defense and asexual reproduction, whereas medusa is focused on sexual reproduction. And let us see, this is the cnidaria, and the bottom part of the star-like part is called as epidermis, and these external light violet color layer is called mesoglea, which is present between this external layer and middle layer. The area present between these external layer and internal layer is called as mesoglea, and these orange color layer, this middle part is called as gastrodermis, and the middle part is called gastrovascular cavity, which helps for the digestion. And between the gastrovascular cavity and epidermis, we can observe the nerve net, which is observed the structure of this nerve net is like net. And at the top of the cnidaria, we can observe the mouth region. From this mouth, it can take the food through the into the gastrovascular cavity, and these finger-like projections is called as tentacles. Next, these are corals. Coral is a class of colonial animal that is related to hydroids, jellyfish and sea anemones. Stony corals, a type of coral characterized by their hard skeleton, are the bedrock of the reef. Stony coral colonies are composed of hundreds of thousands of individual living polyps. Corals are animals. Corals can be fluorescent. Corals eat plankton or small fish. There are hundreds of coral species of all corals, shapes and sizes. Corals can move. Corals support 25% of ocean life. Climate change is the biggest threat to corals. Next, affinities to Ctenophora. Ctenophora shows several affinities with Cnidaria. Some of them are the radial and tetrameral symmetry of the body like the medusa of the hydrozoans. Arrangement of parts along oral aboral axis. Presence of branching gastrovascular canals and absence of coelom. Presence of tentacles and statocyst. Thank you.